Hi, everyone. I've got a very quick and exciting announcement here. We're hosting our very first ever in-person meetup on November 7th from 6 p.m. to 8 p.m. in Denver at the ASHG conference. It's going to be a fantastic opportunity for past guests, future guests, listeners, and friends of the show to come together, share ideas, and chat about all things genetics and precision medicine. We have reserved a very cool venue. We're going to have free food and drinks, and there's going to be a lot of interesting people in attendance. We're sharing this event first with listeners of the podcast as well as past guests. So if you can be there, please register as soon as possible because the spaces are limited. You can register at lu.ma/geneticspodASHG, all lowercase, no spaces or dashes in any of the words. The link to register is also in the show notes and it's easy to use on your phone. Again, that's lu.ma/geneticspodASHG, all lowercase. Mark your calendars, come say hi, and I can't wait to meet many of you in person. One of the challenges of podcasting is you often have absolutely no clue who the listeners are on the other end, except those of you who I really appreciate who drop me an email or say something on LinkedIn. But that's actually a very small minority of people. So if you can't make this meetup for any reason, but you really like the idea, then please don't hesitate to drop me an email at patrick@sonogenetics.com just to say hi. Let us know where you're based so we can keep it in mind for future meetups. And if people like this sort of thing, then we're going to plan to do more in the future. So thank you again, and we look forward to seeing you there. Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Paul Wille, who is the Director of Product Development at Abeona Therapeutics, where he's focused on the development of gene therapies and AAVs in particular. And we're going to, in particular, focus on eye disorders, but more broadly, the role of AAV in gene therapy and what this technique for delivery and this approach might hold for the future and what we've learned about it over the past decades since it's been discovered. So Paul, thank you so much for joining us and looking forward to having the discussion.
Yeah, thanks for having me on, Patrick. I appreciate any chance to talk about viral vectors. AAV is really unique, and then there's been so many traumatic ups and downs during its adoption. I know you warned me here before the show that I don't have to go into too much detail about the basics of AAV biology, but it helps us talk about sort of the benefits and the limitations of this technique. So this is a naturally occurring virus, and it's odd in the single-stranded DNA, and it's not enveloped. And then it requires adenovirus in the wild in order to actually replicate. And so, there's not a very many viruses that are sort of dependent on other viruses to replicate like that. Um, the benefit of it, there's a lot of benefits of it for gene therapy, but one of them, just from the genetic point of view, is that the entire genome essentially can be taken out, and just what's remaining is the terminal repeats, and that's how we're able to put in sort of anything we want into AAV as a vector, which is a benefit over something like a retro or lentivirus, which still needs like the packaging sequence, and how you mess with that remaining bit of sequence so that it doesn't make something you don't want can be a challenge. And
Right.
people have done a great job at that over the years, but AAV is unique. You just need those ends. And then naturally occurring versions of a capsid, the surface of the virus, are capable of binding to a variety of different cell types. And we can alter that specificity through mutagenesis to skew the capsid's tropism, and that's really obviously ideal for a viral vector. and then once inside the cell, the genome, when it gets to the nucleus, becomes an extra chromosomal episome. And so in non-dividing cells, you're able to sort of keep making whatever your gene of
Mm.
interest is through the life of the cell. And so all these factors together, I think, make it attractive, for broad therapeutic and safety reasons.
Yeah.
So for example, we had, a couple of the first approved therapies use natural serotypes. Zolgensma used AAV9, and this is for the treatment of spinal muscular atrophy, where it delivers the SMN1 gene to motor neurons. And then AAV2 is used for Luxturna, which treats inherited retinal dystrophy. So, I think that the main takeaway here is that this is a great gene therapy vector because it really has a broad and changeable tropism and has that possibility for durability depending on the target cell. And as we've seen, relatively safe.
And when gene therapy developers are thinking about this question of delivery more broadly, what other options are there to consider? Because AAV's been around for more than 50 years and has been
Right.
involved in a number of approved therapies, but there are also other options to consider. Like, what's the kind of high level
Sure.
landscape look like?
Yeah, I think really, for the most part, the field has been limited to AAVs, adenoviruses, and some form of retrovirus, whether it's gamma retroviruses or lentiviruses. There's some herpes viruses that have been used as well, but that's probably been more limited. Um, I think that as things got rolling along and you started to see some efficacy with AAV, that's sort of what led to a lot of the piling on of attempts to use it for different diseases. And the safety profile is really very good. I mean, as you can imagine, being able to use a vector that in the wild doesn't really cause disease um, gives a lot of reassurance early on.
Yeah.
I think it just makes the argument simpler for going in. I think as a virologist, which is my background, you know, a lot of the threats from the other vectors can be overblown. But you want to make things as simple as possible, and you want to limit as many hurdles as you can. And in terms of that kind of safety, AV really sort of hit a lot of good notes that way.
Yeah. Yeah, that makes sense. What are the limitations? Like if you could wave the classic question, if you could wave your magic wand and solve one of the sticky problems that you have, what is it with AAV or in gene therapies more broadly?
Yeah, there's a few things that are challenges to the field. Like I alluded to previously, you have this non-integrated genome, which is beneficial in some cells, but if like terminally differentiated cells like photoreceptors that we're going into in ophthalmology indications. But in dividing cells, it'll dilute out. And
Mm.
so if you're trying to do some type of durable supplementation in a cell, in a tissue, where there is actively dividing cells, you're going to have to face redosing at some point in all likelihood. Um, as well, large genes can be challenging. So, the coding capacity of AAV is about 4.7 kilobases. And, that gives you a lot of space for a lot of different therapies, but there are certainly a lot of indications that's not the case. And in that case, you need a multi-partite approach. So you're using multiple vectors, which is something that we're working on with one of our therapies. And we're confident that it can work, but just adds another layer of complexity. And then, depending on the capsid, if you're doing a systemic administration, a very large percentage of the vector gets caught up in the liver, sort of first pass through. And so, this really creates both safety and efficacy challenges. Um, for us, this is, an advantage of dosing in the eye is that you just need a very small amount of vector and it's locally applied. And so, you get around some of those issues. I can talk about that a little bit later. But one of the other main challenges here with AAV is really manufacturing.
Mm.
Um, you know, some of the systemic administrations require an absurd amount of vector, even for a young child on the systemic administration. Sometimes we're using these massive bioreactors, hundreds of liters scale, and this is just for a single patient, and so this has been manageable, I think, for rare diseases, as we get the ball rolling here. But it's obviously unsustainable in terms of cost and capacity. And then as we move into sort of larger population indications. So I think that, great strides have been made to improve the efficacy of this product. And because that will be able to dramatically reduce, the volume and the cost of manufacturing. The causes of these challenges, I think, are complex in terms of both the toxicities associated with systemic administration. But, I think that we're overcoming these challenges through changes in dose, route of administration, improvements in purity and manufacturing. And so I think the future remains bright for AAV vector therapies, despite some of these challenges.
Yeah. On that final point about needing these giant bioreactors, why is it? Is it because of the point you made about so much of the vector just not making it to where it needs to be, or is there some inefficiency in the reactors themselves? That seems crazy.
Yeah. So, manufacturing, it's kind of all of what you said there in
Yeah.
terms of, it's the dose that's necessary, especially in a systemic administration. If 90% of a vector is getting caught in the liver, then you're going to have to make 10 times more go into people in order to get the same kind of therapeutic benefit throughout the rest of the body, depending on your target. And the challenge is really that the production per cell, based on what we're doing right now in these suspension processes, at least initially, was low compared to the amount that you have to put into a person to see efficacy. And one of the big improvements that's coming along is improvement at the cell line, in the upstream portion of the production. And so what we're seeing there is going from what's initially like 10 to the 13 viral genomes per liter of production up to 10 to the 15th possibly. And so you get that somewhere in a 10 to 100-fold improvement there. Obviously, it's going to dramatically drop the amount of manufacturing volume that you'll have to use. So that scale of improvement, it really will change the game. Does it get you into large population indications? We'll see. I think that's still yet to be shown, but it certainly makes that a lot possible.
Yeah. You mentioned the eye earlier as one of the organ systems that is quite amenable to an AAV based approach because it's non-dividing, and so you get this, you get potentially a one-time or very infrequent dosing. Maybe you could talk a little bit about the diseases you work on, and just as a field as
Yeah.
a whole, where are we when it comes to gene therapies in the eye, and then what's to come on the horizon?
Yeah. So I think I'll start with this sort of second part and then lead into our therapies. The benefit of working in the eye, like I mentioned, is that the administration is local. You're using somewhere in the order of 3 or 4 logs less virus because the space going to is so small. And you have a lot of different diseases that have well-characterized single gene mutations that we believe are correctable through augmentation or supplementation with a gene therapy. And we know that AAV gets into a broad variety of cell types within the retina. So the combination of all of those things really makes it an attractive target. And that's not even to mention safety. So, obviously a local administration is better just because it's limited in where you're going. But, there's a little bit of protection of the retina from the immune system. It's not quite like the blood-brain barrier so much, but there is a little bit of protection there. And, and so depending on the route of administration, you're just going to have a lot less limited effects in terms of inflammation. We do see that, but it makes it a lot more amenable to this type of therapy. And so because of all those reasons, we thought it was attractive to move into ophthalmology, and so we're working on 3 inherited retinal dystrophies: X-linked retinoschisis, Stargardt disease, and autosomal dominant optic atrophy. And one big emphasis for going after these indications was the development of a proprietary capsid that we use called AAV204, which in preclinical studies demonstrated in multiple routes of administration in the eye was very good from our data. And we have this capsid came from a license that we had with University of North Carolina at Chapel Hill. And so, like I mentioned, I think that we went into these indications, and AAV is a good vector for them, because it allows us to do this gene supplementation, in a way that it gets to the root cause of the disease, and so we can take advantage of the ability of AAVs to go into multiple cell types with a few of these indications. And in particular, AAV204 has been good for getting into the photoreceptor cells. And this has been important in our X-linked retinoschisis therapy. But I think one real great advantage here of AAV as well is that you can use the same genomic plasmid to package vectors with different capsids. So the ends of the genome, the ITRs, the inverted terminal repeats, typically are in all of these constructs that people use are really mostly the same, and they get recognized by a variety of different capsid serotypes. And so you can go in with the same genome and test a bunch of different capsids and see what's the best for your program. And so that I think that as well is something that's challenging with other viral vectors, where you're putting on a surface glycoprotein that you really only have a few choices, and typically people are using things that just kind of go in everything. And this can kind of fine tune a little bit better, which I think is really optimal.
With each of those, oh, sorry, go ahead.
Go ahead.
I was going to ask what with each of those, I think you're probably in preclinical stage with
Yeah.
most of those today. What does that next step into the clinic look like?
Yeah. So we presented data, proof of concept data for all 3 of these programs at ASGCT in 2023. With our X-linked retinoschisis therapy, and that one's just a little bit farther along than the other 2. What we showed is when we got just the right expression of RS1 in mouse retina. So we've been doing mouse models of the disease with all 3 of these indications. When we show a good expression of the protein RS1, just the causative gene, we prevented the phenotype, which is in this case a separation of the layers of the retina. So RS1 essentially is a glue within the photoreceptor layer in the middle of the retina to keep it together. And so what happens in the disease is that when you have this separation, eventually it just throws off the homeostasis of the retina, and the photoreceptor layer begins to die. And when that happens, then of course you get progressive blindness. But restoring the gene really keeps these layers together. And so what we've shown by using AAV204 as our capsid is that we can apply it in these mouse retinas and get this RS1 expression sort of broadly throughout the retina. It seems to prevent the phenotype from occurring.
That's amazing.
And we also use the same capsid, the proprietary capsid AAV204, in our autosomal dominant optic atrophy program. And in that case too, we were able to show the expression of a causative gene OPA1, but in different cell types. So in this
Yeah.
case, this is caused by changes in the retinal ganglion cells on the inner layer of the retina. And uh, we've shown in other studies in non-human primates using GFP as a marker that these cells are very well transduced, and that you can see this GFP kind of spread out to the periphery in these cells, even though the dose went into an area just in between the macula, which is central vision, and the optic nerve. So I think because of that, it really provided sort of an ideal vector for trying to get into these cells. In terms of proof of concept, that's coming along nicely, but we still have ways to go there.
And in each of these cases, generally speaking, is it a like an either an X-linked or autosomal dominant loss of function? So there's, it's a haploinsufficient gene. You need two copies, but they only have one, and you're coming in with or is it recessive or is it combination of the two?
Yeah, so for X-linked retinoschisis, because it's X-linked, this essentially you really only see the phenotype in males, and then
Hmm.
just missing the one copy on the X chromosome. In the case of autosomal dominant optic atrophy, it's an imbalance, and what our strategy is really is supplementation in this case because the data that we've seen and you can see in literature is that this addition of wild type OPA1 is sufficient to sort of move the balance back towards homeostasis. That's not going to be the case for a lot of the dominant diseases where they really so truly prevent the wild type copy from functioning. But in this case, the supplementation sort of swamps out the effect of the mutant form.
And what does the universe of monogenic eye diseases look like? Like, are we just kind of scratching the surface with what we're talking about, or are the ones that you're focused on some of the most common amongst these, still relatively rare diseases?
Right. Yeah. No, it's a great question. Yeah, there are a wide variety in the order of hundreds of rare diseases, of monogenic rare diseases within the eye that primarily affect the retina, and um, we have stuck to ones that are sort of on the less rare side. This is a real challenge in the field right now because these ultra rare indications that cover most of these kind of monogenic diseases are really hard for sponsors to follow up on because economics are just not there for us, and there's a lot of causes to that, but you know, and we've talked about one, right? We talked about the fact that the manufacturing is so challenging and so cost intensive, but um, it's a lot more than that. You know, it's the whole path, it's a whole development pathway. Because
Yeah.
regardless of how many patients you have, if you have uh 10,000 patients or one patient, the regulatory pathway is the same and the costs associated with it are the same. There's increasing flexibility uh, for that from agencies like the FDA in order to help a little bit with these rare disease programs. But even in the face of that sort of slow evolution, it's just not viable for industry sponsors to go into these ultra rare indications. So, I think all of us hope that as we go along and make improvements in things like manufacturing, use platform indications like the FDA has talked about, that we're able to use these sort of less rare indications as that sort of proof principle that we can consistently, change the course of these diseases with AAV, and then use that to move into rare diseases over time.
Yeah, yeah, it makes sense. It's a tricky problem, but it is, to your point, nice to see there appears to be more and more regulatory innovation actually in thinking about how do we solve this problem in a way that still protects patients and puts safety at the top of the list, but makes it either more attractive to invest or easier to to run one trial instead of two, for example. So I think like I'm optimistic, but for the ultra rarest of diseases, there's still a lot of hoops to jump through.
Right. Yeah. And you know, this is at this moment, the partnerships between government, industry, and academia that are starting to come online, like the Bespoke Gene Therapy Consortium, give you a little bit more of a sense that this is a tackleable problem, but it's really going to take, I think, equal effort on all ends. And so, when like Peter Marks, the CBER chief, talked about regulatory flexibility, we need to really refine what that means over time. Again, ensuring safety, that's obviously number one. But making sure that we are set up to do things, that allow us to take advantage of the learnings that we've had up to this point, in these rare indications.
You spent a lot of your career in academic research actually, and then moved into industry. I'd love to hear a little bit about what spurred that change and also how you found that transition to be.
Yeah, yeah. So I started my career in a lab at the University of Washington right after undergrad that was studying the role of inflammation in atherosclerosis. And so I started with an interest in immunology, and these are macrophage dependent processes, and got to know and love the macrophage in all its complexity. And after a couple of years there, I went to grad school down at HSU in Portland, Oregon, and took a virology. I had always had an interest in sort of the scariest of the diseases, and had that vision of like somebody going into the class 4 labs and
Yeah.
studying blood or something, right? Yeah, instead I ended up in herpes virus. So, but at that time, what that started and what's continued in my career is this interest in viruses as vectors. And uh, I think that the possibilities really for all of these different kinds of viruses, that we haven't really tapped into the potential of them as much as we could yet. Um, and so I had my career in grad school studying herpes viruses, surface glycoproteins, the cytomegalovirus, and had a couple of interesting papers about one in particular about how we could put the glycoproteins that are the receptor binders in one cell, and then put the fusion protein on a virus that's missing receptor binding proteins, and cause the virus to fuse into the cells, showing this like weird flexibility in the use of these glycoproteins. And it's those kind of learnings about how viruses get in the cells, which I think is something that is really important in
Mhm.
understanding how we can manipulate viruses for the use as vectors. So after I got done with grad school, I moved here to Cleveland, and started in a lab studying HIV. I joke that my mentor at the time, like what aspect of HIV is kind of like everything.
Yeah.
But again, I focused on something relevant to the use of lentiviruses as a viral vector, and we had characterized in this lab sort of a novel packaging element that seemed to be separate from the standard psi sequence in HIV, and trying to understand sort of how we could manipulate that to either improve packaging and viral vectors. After that, I spent a little time in another lab at Case Western Reserve University, prior to coming here to Abeona. And, the transition for me just made a lot of sense because, um, here's a company that's working at the forefront of AAV and viral vectors. And, over the years, it's not like I focused on one type of cell or one type of tissue or whatever. I could come in and say, okay, what disease should I be working on? Let's see how we can use these vectors in a way that really um, gives us the best possible chance of treating whatever disease it is. And so, yeah, I've been here almost 7 years now, and it's I think it was the right move for me at the time.
Yeah, yeah, definitely. It is in some ways like the worlds are, especially if you're doing research, are very similar, right? You get to work on interesting problems, you get to work with great people. Maybe you don't get to publish until until you get full approvals and the posters get approved by legal and that sort of thing. But otherwise,
Right, right, yeah.
there's a more porous barriers than ever before between academia and industry, right? For almost everybody.
Right. Yeah, and I think it's what's great about it is that it goes both ways because, you know, uh, traditionally, right, there was just this wall, and it
Yeah.
was industry people did their thing and academic people did their thing, and there was some learnings that kind of like got thrown over the wall, but um, they were just really very separate. And then I think you had a period of time where industry can see and start taking things from academia to move forward. And now I think it's really more bidirectional where obviously sponsors like us are licensing in programs from academia, but that so much of the academic setup now is comes from a lot of learnings from industry in terms of commercialization of things or moving things into clinical studies and getting them pretty far along before they find that they need a partnership. And so I think that those changes really, and also the mindset change of like, you're not going to the dark side if you
Yeah.
transition from academia into industry, right? That mindset I think completely changed, and because of that, I think that talent goes to the right place better. You know,
Yeah.
not everybody is meant to be in academia, and not everybody is meant to be in industry. So, a lot of it comes down to personality and interest. And, as we broaden our view within graduate programs and postdocs and things that happen at universities for where those careers can go, it really, I think, allows the opportunity for people to flourish in the right place for them.
Yeah. As we wrap up here, I'm interested to ask you about an area of precision medicine that you're really excited about that may not be quite on the radar of the mass market yet. Is there anything
Mm-hmm.
that you have in mind that you think could be really interesting a couple of years down the road, but maybe isn't on everybody's radar yet?
Yeah, it's a great question. I think there's a challenge here in making sure that we don't just strive after the new shiny thing, right? So I think that sometimes we have a tendency as new technologies come along to be a little too aggressive into them without taking into account possible limitations. And so just to start, I think that we don't want to throw out the baby with the bathwater here, and that there's a lot of possibilities for AAV still. And it's not just gene supplementation in monogenic diseases. I think that there's a possibility for a strong role of AAV in delivery of gene editors. And you're seeing that. You're seeing CRISPR, you're seeing that with the base and prime editors. That's just barely been touched. And I think there's a lot still there. I think the future for lipid nanoparticles also is very
Mm.
good. And I think that there's a lot there that we haven't even begun to think about. The delivery of RNA obviously is very important and will have a great place in a lot of different indications. But, in the simplest terms, it's just a lipid covering on whatever you can do, right? And so I think there's a lot of possibilities there. And so, I think that's one place where there's going to be an evolution of how we use some of these. But I also think that we'll find limitation that, we just haven't yet. And that will spur that sort of iterative process that we went through with AAV. People are like throwing money at every company that had some new indication and great proof of concept data with AAVs, and without really realizing what the limitations were, because we just hadn't run into them yet. And right. I think there's too many people that once those limitations come up, they change directions as quick as possible. And I just don't think that matches up with what we're seeing, not just us at Abeona, but just the field in general, about how we're able to keep plugging along and really think that we're going to get across the finish line with a lot of these AAV vector therapies.
Yeah, it makes sense. Well, Paul, I just want to say thank you for taking the time to share all this with us. I learned a ton about AAV, actually. It's something that I hear about almost every day, but rarely have the opportunity to really go deep into the subject. And it's clear that you have spent a lot of time thinking about it. And I'm pretty convinced that it's something I should spend a little bit more time thinking about as well. It's one of those things that has been around for a really long time. And so it's easy, to your point, to kind of take it for granted or get attached to the shiny new object, but it has had staying power for a reason, it sounds like.
Yeah, I totally think so. Absolutely.
Right. Well, thank you. And thanks everybody for listening to us as always.
Yeah. Thanks for having me on, Patrick.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.